Okay, welcome to another session of the Bioinformatics and Genomics from Data Analysis to AI Applications workshop series. I am Michele Cosi. I am an educator for the Data Science Institute. My role in the past has been the one of bioinformatician, where I would align reads in order to contigs in order to make genomes for studying. Today is a topic that is a little bit outside my knowledge pool, and I would like to cover or give you a beginner's guide to RNA-seq with DESeq2. Let me share the right screen. I believe it's this one. You are now seeing, yes, okay, cool. We're in the right place. Okay, I'm going to share this GitHub link to you folks in the chat. This is where we have the list of all of our workshops for the bioinformatics series. If you were to scroll down, you can access each workshop's material here on the right. We are currently working on getting those recordings up and running. We are running a little bit late on that at the moment. For today's material, you can scroll down to the 0206, a beginner's guide to RNA-seq with DESeq2, and you can open up the material by clicking on it. It has to be mentioned that we're going to be sort of continuing working with the data that we have worked with last week. And we are going to bring in, I think, 3 more controls and 4 different experimental four experiment type data. So today, the idea will be to cover a little bit of what happened last week, what we did, how we prepare our data before jumping onto R. Because I do not want to spend the entirety of today's lesson preparing the data, I have created and prepared the data for you guys if you do want to follow along, as well as installing DESeq2 and all of the libraries that it requires to run it on an application on Cyverse that we can use together in order to obtain some visuals from the differential expression analysis. If this is the first time for you to be here, the way that this is structured is that we use GitHub Wiki, and I essentially just use it as visual aid. All of the material that I am going to cover, it's here, but I'm not going to go line by line. This is for you to access in your own time and to just for all of us to keep track of where we are and some of the material that we may want to cover. Today's structured in 3 main blocks. One is introduction to differential expression analysis. The second is talking a little bit about experimental design and what we did last week. And then we're gonna go into Cyverse again and look at a script that I have written or helped write in order to visualize the data that we are working with today. Once again, this is not an introduction to computing. My understanding is that you have a little bit of knowledge on the terminal or using R or Python, and you do have a little bit of background on in bioinformatics, or at least know a little bit of genetics and genomics. The expected outcomes of today is to have a general understanding of differential expression analysis and its workflow, revisit what we did last week and give you a basic understanding of how DESeq2 works. So let me move some Zoom things around. Oof. Off. There we go. This is better. Okay, so this workshop focuses on RNA sequencing analysis and differential expression analysis, more specifically, and looking at the DESeq2 simply because it is accessible, or I found it accessible. Next week, we are going to have a couple of experts. Next week and the week after, we're going to have a couple of experts that are going to go into differential expression analysis and RNA-seq even in more depth. So this is going to be the introduction to what is going to be a much deeper discussion over the next 2 weeks. So for today, right after running DESeq2, we're going to be using ggplot2 and enhanced volcano, as we are going to create histograms and a volcano plot that you may want to, or I learned how to read whilst creating the materials. So I may stumble on myself, but please let me know if something does not work for you. So last thing that I did mention last week as well, but I'm going to mention today again, is that we have run this workshop over the course of 3 hours together with the Texas A&M HPC group called ACEs. So this material is very much getting cut or shortened down in a more size that you can digest quicker, but it will it will not go over specific aspects. You can always reach out to me in your own time, and I'll try to answer as much as I can. Today's experiment is revolving around the house mouse, Musmunculus. And the RNA sequencing data was given to us by the Texas A&M, but aligned and trained by us. And I'm going to cover the techniques that we used, which are the same that we used last week as well. Okay, that was a mouthful, a good 10 minutes. But let's talk about it. What is differential expression analysis? Well, let's take a moment to try and answer it in human terms. We all know that the genome and the DNA is quote unquote fixed. That's where our code relies, but our code may change depending, or the expression of our code may change depending on whether it is too hot, so the weather, or too cold, or whether you're going through something in life, a very stressful situation, for example, or you're just being happy. So there is different expressions of the genome that happen at different types of our lives, and it is up to us humans to study how this works and which genes are expressed and which genes are down-regulated, for example, when something occurs. So my understanding of differential gene expression analysis is exactly that. We are trying to understand when we are put under a specific stressor, if a specific set of genes is being expressed or down-regulated. The outcome of DEA can give us some insights on unobserved phenotypes. When I was a student, we were running an experiment in the middle of the desert to see how the lack of water would stress a specific plant. In that case was sorghum. And we would not only have to keep an eye on the plant for various phenotypic changes, but also we had to do RNA sequencing to see how the plant was responding to the lack of water itself. If you do have a question about anything, feel free to raise your hand. I do not want to minimize anyone, and I want to make sure that everybody has their questions answered. So why is DA important? Well, it helps us understand how genes function in different situations. So in this specific case, given if a gene is active during cancer but not in normal cells, it might be important for the cancer cell, and this will give us a little bit of an understanding on the function of this specific gene down the line for a potential treatment. What are the big steps of DEA? Well, we are looking at 4 main steps. The first being the measuring the expression. So RNA sequencing does provide us a small snapshot of gene activity by counting how many RNA molecules or reads, I don't know why I said molecules in this specific case, I mean reads, correspond to each gene in a specific sample. This read count reflects the gene's expression level in each sample. So let's say that we do have 10 people, and they are in this experiment. We want to run RNA sequencing. We want to sequence their RNA to see how differentially expressed specific genes are under a specific stressor. Obviously, then we compare with the control groups. We then identify the differences. So we take the RNA sequencing data and we want to figure out whether there are differences between all of these experiments, between all of the people that we're getting the RNA-seq data from. But then we want to do some statistics. We want to figure out whether this data is meaningful or it is just something that we are observing. And then we want to interpret something called a fold change. And a fold change is something that still today I am getting to learn. So from my understanding, I've written this down as I understand it. If a gene shows a large fold change with a low p-value, it's considered differentially expressed because its expression is significantly different between the conditions. So I understand this in words, meaning that if a specific gene has a low p-value, then it is significant. Okay. And how do we observe this fold change? We use something called a volcano plot. And you may have seen something like this in publications where you observe specific genes that are further and further away from the center of your graph. Now, we were talking about a p-value and a specific fold change that we want to respect at the moment. The fold change in this specific case, I believe it being the line right, well, this is the p-value, so we want the p-value to be a specific value, but then the fold change that we do observe is visualized as the dots that are further and further away, and that is those are the ones that we want to keep in mind. This is a really bad explanation, but if I were to bring it back to something easy to understand, look at the dots that are further away from 0.0. Those are the ones that we need to keep in mind. Is there a gold standard threshold setting in the aspects of a bioinformatician? I will say I'm not sure if there is a gold standard. From my understanding, it's that it varies between samples themselves. I do tend to mention that you should try things out on your own first, but this is something that I am not able to answer you. I can get back to you later, or Google may help you. Apologies, this is not necessarily my field. I just am trying my best to educate folks in it. No worries. Jose asked, the differential expression ones are under the threshold. The differential expression ones are above the threshold. Or, hold on. The differentially expressed ones, let me read this again. Yes, so the differential expressed ones are above the threshold. So what you're looking at is the threshold being here. You want to observe the ones that are both further away from the center and further away from the threshold. So yes, these ones right here. No problem. I just, thank you for asking. I'm also trying to catch myself saying something wrong here. So thank you for asking the questions. Okay, so, but there is a higher p-value above the threshold, so, and we need low p-values for significance, right? We want a low p-value. Okay, hold on. Let me get this straight. We want a low p-value. And I think what you are observing here is the log 10 fold change. So the higher it goes, the lower the p-value is. Yes, exactly. Yeah. So yeah, I did forget to mention that this is what a volcano plot does. It helps with a visual understanding. So yeah, do keep in mind that this is a negative log 10. So thank you for pointing that out. Okay, so for the ones that were not here last time around, I do need to recap what has happened. We used 5 different tools in order to prepare our alignments. And these are FastQC, TrimGalore, HiSet2, which index the genomes and aligns the genomes, SAMtools, which sorts and index alignments, and DESeq, which counts specific features within the gene, or specific features within the reads. There we go. So first of all, we have FastQC, and if you are new to FastQC, what it does is that it gives you the quality of a specific read, and these are ideally the raw reads that you get from your Illumina. What we were doing last week is that we were observing and talking about what FastQC does and how to read FastQC data. And in this specific case, you can see that most of the reads are in the green, but we do want to trim some of the longer tails over here. For that, we use Trim Galore. I always catch myself saying Trimomatic at that point. Trim Galore, we use the standard settings on Trim Galore, cutting every read that's below 20 reads long, and we have improved the quality of our reads. In short, this is something that I said last time around as well. What you want is to have most of your reads above the green or within the green line. This sets a Phred score between 28 and 40. I think it changes a little bit between the type of reads that you might be using, but you always want to be within the green. So this is actually good for us. Fast forward. We then use the tool called DESeq2 to first build a genome index. And for that, we use DESeq2. And then we use DESeq2 to align the trimmed reads onto the index genome that we have created. We ended up getting a 93.52% overall alignment rate, which is very good. I did receive a question from, I do not want to get your name wrong. Are you here? No, you're not. I did receive a question from one of the folks that attended last week's seminar regarding, hey, when I run things with DESeq2, the alignment of DESeq2, I get different answers in my alignment depending on how many cores I use in order to align my my reads. That was actually quite interesting, and I had to look a little bit into that. In this specific example, I was aligning things using 2 cores. -P is the flag for how many threads you want to run, and 2 is the set of cores, which yielded a pretty good alignment overall. But if I were to higher the number of cores available, maybe you wouldn't see 197,200. Maybe you will see 197 full stop with 200 not really aligning either here or somewhere else, but not aligning full stop. But the total percentage of alignments is actually still almost the same. There was, I think, a 0.007% of difference between the 3 tests that were run. So in the grand scheme of things, changing how many threads you use can lead to very minuscule changes. And with these very minuscule changes, unlikely this will be important to your research. However, you can be quite meticulous and enforce that you always use the same number of cores when you're running your alignment. So, with that said, why does this happen? Simply speaking, let's assume that you have an 8-core machine. An 8-core machine means that each core, so 8 cores, is able to take some work. What DESeq2 does is that it groups the reads that need to be aligned into X many groups. And in this specific case, we have 8 cores. So DESeq2 will create 8 different groups. And there may be some reads that would benefit from being on a different group. And therefore, the alignment still happens. The percentage will still be somewhat the same. But the alignment will be somewhat the same. However, the change will be extremely small. But again, it is up to you to define if that minuscule change, it's important to you or not. So thank you for the question. I think thank you, June, for the question in the mail. I felt the need to refer the same questions to everyone else. So going back to what we're talking about today, DESeq2 aligned the reads at this point with a pretty good alignment rate. We then use SAM tools to sort, to index and sort our alignment. What this does is that it allows for downstream analysis to be performed in a quicker manner. Last, what we did for this specific experiment, we used the DESeq count in order to find out a couple of things. The first is how many genes there are in our alignments and their position. If you remember, last week we ended with a text file, a really long text file with all of the potential, all of the genes that were present and their position. And I said, don't open it because otherwise you won't be able to read it and things are going to break. Or whatever I said, I'm not really sure. But I needed to go through what we did last week and give you a little bit of understanding on the fold change before we jump into DESeq2, using DESeq2. So DESeq2 is an R package. I think the bioinformatics community is somewhat used to DESeq2 at this point, or at least using R. So DESeq2 is an R package that's used for RNA sequencing data, and it does take in different types of input data. It can go from raw counts, for example, the ones that such as the one that we have generated last week. What it can do is that it can take the data, it can normalize it, it can create models, and it can do some statistical analysis as well. However, what we're going to do today is that we're going to use ggplot and Hansel-Volcano to create our images, which is what we want to do as scientists in order to understand our own data. What we're going to be doing is that we're going to be doing the log2 fold change, and we're going to adjust the p-value. We're going to get the p-value and adjust it, essentially removing everything that is below a threshold, and we're going to create 3 different images. The last thing that I do want to mention about DESeq2 is that if you are running it on your own, you need to understand the experiment before you run DESeq2. Because, and why? Because DESeq2 takes into consideration the design of the experiment that you are running. For example, here are 4 examples, but what we're going to be doing today is that we're going to have a single factor design where we have a treated versus a control. And in DESeq2, you need to use very specific ways to tell DESeq2, hey, I did a treated versus control, a single factor design, and this is how you mention it. This is how DESeq2 will ingest that information within the command line, or within the line of command. So the last thing that you will write is design equal, and how you're going to write your experimental design. It can take 2-factor design, so a condition and a batch that can be independent are independent from one another. It can take an interaction model or a multi-factor design. There is more, but I simply don't have neither the time nor the knowledge of knowing all of the designs for DESeq2. Simply speaking, this is something that you need to keep in mind when you are starting your experiment. Even before doing the sequencing, you want to make sure that DESeq2 will be able to read your own experiment design. So With all of that said, I wanted to mention that if you are wanting to do this on your computer or just want to look at the code, you can click over here, and this is the code that we're going to run. I've done as much as I can to add comments on what each line does. If you are following along, then at this point, you know that, well, we are going to be using Cypress for this. I have all of the information required for you to run this through Cypress, so I'm going to go ahead and show you how. First, we're going to go to Cypress and we're going to log in. I have already done so, and I would like to access the app that I require, which is called the Jupyter Lab Bioscience with RStudio. I can click on it, and this is the version that I want to use, 3.6.3 BioSci RStudio 441, and I want to click Next. For CPU cores, I'm going to ask to use at least 8 with 16 gigs of RAM, and then I'm going to click Next, and I'm going to launch my analysis. This shouldn't take too long. You can see that now my analysis has been submitted, and soon it will start running. I guess the question is, why are we using Cypress, and also why are we using an app on Cypress? Really simply speaking, I don't need to show you now all of the apps or all of the software that I've had to install to make this work, and you don't have to install to make this work. All of this software has been containerized into a single tool that you are now seeing as Jupyter Lab Bioscience with RStudio. I also am not running this on my computer because it will bog everything down. Also, my computer, I don't think it has 16 CPU cores, so, or I did, yeah. By all means, this is going to make my life a lot easier. I think it's still submitted. I'm going to click Go to Analysis. I want to see if this is starting. It's downloading my containers. Okay. Wonder if it's stuck somewhere. I'm going to click More Actions and relaunch. 8 and 16. Launch analysis. I'm also going to click Go to Analysis. Let's see. Last week it was very snappy. This week is complaining a little bit. And I tested things out earlier today. They were a lot quicker. I'm going to give it another minute. If it does not start within the minute, then I already have a running app that I can use for my own presentation. If you are following along, I hope that your app is already up and running. Otherwise, this is a good time for questions if you have any. Ooh, okay, hold on. I have a question related to trim galore, but it has to do with RRBS FASTQ files. They have a RRBS flag, but I'm not sure if I should use it, as I don't know if my data was digested using MSLP, which is prereq in the trim galore documentation. I'm not familiar with this pipeline. If anyone could tell me where I should find how the samples were digested, or if the sequencing machine type used. Okay, will tell me how it is digested. I believe that I was an Illumina HiSec 2000. This is also public data. Thank you. I wonder, this might be a stupid comment, but have you looked at whether there are publications that have something similar to your experimental setup, Jose? And if there is publications that have a similar experimental setup, do they also explain what they did with their trims? Maybe you're typing, but by all means, this is the first thing I will do. See if there is a paper out there that has done something similar or to what I have to do. And whilst you are typing, it seems like things are not starting. So what I'm gonna do is that I'm going to fall back on an analysis that I had running. Okay, I'm not involved in the experimental design. I am just doing the data analysis, but I can work with the others to find out. Yes, please do. I think that will be able to point you out in the right direction. Okay, I'm gonna delete the folder simply because I want to be in the same level as you folks. I don't care if it's not empty. I'm gonna load it again. I'm gonna go to my terminal. And the reason why I'm going to the terminal right now is because I need to download something. I need to download a specific folder. And since there already is a folder with that name, I'm gonna rename it tutorial old. Do not do this, you don't need to do this. And then I'm gonna go cmd init, put in my name, Così Michele, and my password. And then I can go ahead and paste the line that I had just copied from. What did I do? From today, which is this one right here. Go to get cmd -- progress and download this folder. Seems like I am done downloading the folder, and I can go ahead and open it, look a little bit about what is inside. I see a counts, which is actually where all of our counts exist. So I'm not giving you all of the all of the raw data. I'm just giving you the counts that we need for the next phase. And I have an R script called DESeq2_tut. What I'm gonna do now is that I'm gonna press plus in the Jupyter lab and click on RStudio. I'm gonna close this and I'm gonna, Zoom is in the way. I'm gonna clean everything up. Okay, I'm gonna clean everything up. And I'm gonna go ahead and open data store, RTutorial, and DESeq2. Now, ideally, you shouldn't have anything here, and it should look something like this if you have a running RStudio like me or an application. Man, they're still not running. Interesting. Okay. All right. Oh, well. So what we're going to do now, we're going to load a couple of libraries, then we're going to filter the low counts from our data sets. All righty. So I'm going to load ggplot, feedmap, DESeq2, and enhanced volcano. And I'm going to set the working directory in, well, essentially where I am now in the counts folder. So this is basic R. Everything should be pretty straightforward. Next thing we do is we can list the files in case we want to know what they are, but we have already really seen everything that we need. And we can even look into the files using system. But then we can go ahead and load the sample table with the sample metadata and the conditions. What we are doing right here is that we are looking, well, we do have a CSV, and this CSV has the name of the various controls and treatments, and it will be used in the experimental setup. Also, I've not mentioned this before. I should mention this now. We have 4 controls and 4 treatments. These treatments are for NAD, which is a quite important I do not want to say it wrong. I think it is where is my losing my notes, folks. I'm losing my notes. NAD supplements, they were given different NAD supplements with and NAD itself is the nicotinamide adenine. Oh my gosh. Nicotinamide adenine dinucleotide. This has been a really long time since I mentioned it or since I tried to say it, but it is important for the metabolism. So they were given, these are mice which were given different amount of NAD supplements to see, and they want to check how the supplements affects the mice essentially, and they're using differential expression analysis to see this. So we have loaded the data, and now we have to go ahead that it is in the right data frame format. Error. Why am I running into an error? Let's run this one right first. There we go. I did not run it. And yes, it seems like it is able to load it in the data frame format. And we can even look at it if we want. That's exactly what we were expecting from the CSV. So now we can actually go ahead and create our DESeq2 dataset. And the way that it works, it's quite straightforward. You need to use the DESeq dataset from HDSEQ because HDSEQ is the last software or the last tool that we use in order to create these counts. We load in the data frame that we just created in sample data. We want to specify which directory it is in. And as I mentioned before, we do need to go ahead and talk about the design and condition. So we're just going to go ahead and run this. It is a warning message. Don't worry about it. I need to move this over here. It is a warning message, but it still runs. So warning messages are not as errors, nothing breaks, but you need to keep in mind something may not be working as intended. Such as some variables in design formulas are characters converting to factors. It's just telling you what it is doing. So everything is okay. We want to look at it. If we want, we can look at it. It tells you how many rows there are. It can tell you the total dimensions of the file. But otherwise, we are set. There is this many rows and this many samples themselves. At this point, we decide to filter out the genes with low counts. We want to remove everything that has fewer than 10 counts, essentially. So I'm going to go ahead and run this, and I'm going to add this to our data frame. we are updating the data frame to only include the high counted genes and quote unquote high count more than 10. So now that we do have a complete setup, what we did is we ran DESeq2, but before that, we made sure that it is in the right format. And after running DESeq2, we decided to remove the genes that have low counts and update our data frame. So now we can actually go ahead and do differential expression. We're going to run DESeq2 or DESeq, we can call it through DESeq, onto our DDS data frame. And this should take, oh, it's already finished. And then we extract the results from the analysis and put them in res, which can stand for results. And I do think results is part of the DDS framework, DDS function list of functions. So I'm going to click on res to make it look to bring up this little data frame. But what are we looking at? And I feel the need to go through line by line. So here on the column by column, here on the left, you have the base mean, which is the mean of normalized counts for all samples. Then you have your log2 fold change of the NAD supplement versus the control. Obviously, we want to visualize this column because if it is just in digits, we can't really figure out what it says. So, and then we have the long fold change standard error over here. Then we have the wild statistic NAD supplement versus control instead. We have the p-value and the p-adjusted value, or the BH corrected p-values. And we want to now be able to do a couple of things. We want to adjust the p-value or to count only the significantly differentially expressed genes. So we do obviously a p-value that is smaller than 0.5, as we were talking about earlier. And there is 5,453. Okay, we can work with that. We're going to filter these genes. So we're going to keep the significant genes, and we're going to write them onto a CSV. So what we did over here is we ran DESeq2, like DESeq2 function of DESeq2, or the DESeq function of DESeq2. And we decided to keep the genes, the 5,453 genes that we think are significant to the current experiment. So now we can run visualization. The first one is a PCA plot. We are going to do a log transformation. And then we're going to calculate the row variance and select the variable genes, the top variable genes. We're only going to select the first 100. This is also for visuals. I wouldn't call it a standard, but also you want to clean as much in order to make sure that you understand what you're seeing. And then you can go ahead to create your PCA. And PCA stands for Principal Component Analysis. For folks that do not know what PCA is, what essentially it shows you is groups, and are these groups really close or really far from each other? If they're far from each other, then there is significant a significance in the treatments. If they're close to each other, there isn't. So that is overly simplifying things, though. Okay, so in the interest of time, I'm running a little bit behind. I'm just gonna go ahead and run through the calculations and create the PCA analysis. And from what we can see, I'm gonna make this a little bit bigger. There is a difference between control and the treatment. So what we now want to know is, okay, how different? So I'm gonna make this small again. And I'm going to run the next visualization, which is with a volcano plot. To help us, we're going to be using Enhanced Volcano. And I will say to go with these simply because they have worked for me. I will not be hurt if you tell me that these are wrong, but by all means, they have worked for me. So they may work for you in your own experiments. Ooh, this is a little bit claustrophobic. Let's make this a little bit larger. It's gonna reload. There we go. So as we did mention earlier, this is a log 10 fold change. And what we want to see is these ones right here. These ones right here are the genes that we believe are the most important in these specific experiments. These are the ones that are likely that have some sort of magnitude when it comes to, hey, under a specific stress, is the metabolism of this mouse, of this being, different than our control. So these are likely to be the genes that are important for this experiment. Now, it is not possible to point out, oh, CONA4 is the one gene that we're looking for. But CONA4 is potentially going to be one of the genes that we need to keep into consideration. But also, this is good practice for people that are running into the question of how do I create a volcano plot? What does it do? And can you help me create one, for example? The volcano plots can go hand in hand with the heat maps. Once again, I'm zooming through this because we're almost at time. So I'm just going to select all of it and press enter. And what the heat maps will do is that they will tell you if a specific gene in one of our samples, either control or treatments, it's important, essentially, is being upregulated or downregulated depending on the treatment or the control. So in this specific case, we're looking at the warmer colors being genes that are more upregulated, and in blue, the ones that are not, that see no change in the treatment. So on the right side, we have the list of genes, and at the bottom, on the x-axis, we have the list of our samples. And from what I can see here, NAD2 may have some, or these genes here may have something to do in NAD2's treatment. So maybe it is time for us to look into these genes and see if maybe if we knock them out, something during a different experiment involving NAD2 will change. But the reason why I wanted to show you also this R script is not only because I wanted to show you, hey, here is an example. No, here is an experiment that that I can show you is because when I was in my early days of a bioinformatician, one, I did not really understand how to read these graphs or how to read this heat map or the volcano plot. Two, the question is, how do I create it? So after many years, actually, I figured out, well, I didn't figure out, I was able to meet people that would slowly teach me about all of these things. So maybe today, maybe I'm not the greatest at creating these visualizations, but at least I understand what they're trying to tell me. So take that take that however you want. I'm still learning today in this field, and every day there is always something new to learn. So folks, it is 2:57 and we are getting to the very end of the workshop on DESeq2. What we did is we took the data, excuse me, going down. We took the data from a treated and a control house mouse experiment, and we were able to set it or prepare it. We were able to align it to the to the to the genome of the reference genome of the mouse, and we created a PCA plot. We ran some differential expression analysis. We were able to look at a heat map. We created even an enhanced volcano or a volcano plot that you may be able to now try to create in your own laboratory. From last semester, there were a lot of people interested into learning more on RNA sequencing, and that's why next week I'm bringing in a couple of experts in Simona Merlini and Francesca Vitali to explain a little bit in more depth the tools that they use in order to carry out differential expression analysis and more depth and go into more depth on RNA sequencing. With that, I would like to thank you all for your patience and for your, I'm bringing up, come on, you can do this. There we go. I would like to thank you all for today, and I hope to see you again all next week. Take care.